And to finish up that last point, titin is a protein that anchors the thick filaments to the Z line. So here in blue are the thick filaments, and that green zigzaggy structure is the titin protein filament that holds the thick filaments, anchoring them to the Z line there and over on that side there. So during contraction, the sarcomere shortens, and that means the Z line moves closer to that Z line. So the sarcomere will shorten as the thin and thick filaments slide past each other. So with shortening, the degree of overlap of the thin and thick filaments increases. That shortens the sarcomere, and all the sarcomeres that link together in a myofibril shortens. Then the entire muscle fiber shortens, and then the entire muscle in turn will shorten, causing movement. Okay? So let's look at this mechanism. The myofilaments, thick and thin, zooming in a bit here, the thick ones in blue, thin ones in red. The thin filaments are mostly, the ones in red, are mostly actin protein. Zooming in here and yet zooming in again. So the red spheres are the actin subunits of the thin filaments. And there's two regulatory proteins, tropomyosin and troponin. Here in teal is the tropomyosin, and the little spheres here in orange-red are troponin. And we can only see that in this diagram where we're really zoomed in close. At lower magnification, all we see is the red thin filament, indicative of mostly actin. So these two proteins, regulatory proteins, troponin and tropomyosin, are essential for the contraction process. What happens, so here's the thin filament with tropomyosin and troponin. What happens is when calcium gets released from the sarcoplasmic reticulum, and we'll talk about this whole mechanism. When calcium gets released, calcium here in green, binds to troponin in purple in this figure. And when that happens, the tropomyosin shifts, moves, and that exposes binding sites on the actin protein. And those are binding sites for the myosin heads. The myosin heads are on the thick myosin filaments. And these little kind of bumps that you see here sticking up are the myosin heads that will latch on to the myosin binding sites on actin here as soon as calcium floods into the area, latching onto troponin, shifting tropomyosin, exposing the myosin binding sites. So what happens is the thick and thin filaments will form what are called CrossBridging. And that's when the myosin head binds here on actin. And then that causes, as we'll see, the sliding of the thin filament versus the thick filaments. They'll slide past each other. And in doing so, the Z lines will move closer to each other as the amount of overlap increases, shortening the sarcomere, and then the myofibrils, and then the fiber, and then the whole muscle. Okay? So the thick filaments here in blue, if we zoom in closer, they're made up of myosin subunits with a myosin head with this pivoting hinge on them. And it's these things, these myosin heads, that latch on to the actin protein, the actin thin filaments, when calcium is around. So myosin has this globular head and a long tail, and they're kind of like all bound together, like so. So during contraction, the myosin forms what are called cross bridges by binding to actin. And that's possible once calcium binds to troponin. Tropomyosin opens, exposes the binding sites on actin for the myosin heads. So muscle contraction, when the muscle contracts, meaning when the sarcomeres contract, the sarcomeres shorten as the actin slides past the myosin. So we kind of see it here schematically in this figure. There is the Z lines. Notice they're further away apart from each other than we see them here. So that means in the bottom picture, the sarcomere is contracted and over here it's relaxed. Notice that the dark A band stays the same length because all that is the distance spanning the thick myosin filaments. But notice what happens to the I band with contraction. The I band shortens. And why does it shorten? Because the Z discs move closer to the middle M line, so this distance will get shorter in each sarcomere and with the adjacent sarcomeres. So the I band will get shorter during contraction. And notice in the very center, the H band, with full contraction, the H band will essentially disappear. As you see here, very thin versus thicker here. So let's see a little video here on the mechanism of sarcomere and muscle contraction. It should play for you, I hope. There we go. It's a YouTube video. Of course, after the commercial. Our volume looks okay.
When a muscle contracts, the actin filaments within a sarcomere slide along the myosin filaments. It presents a series of sarcomeres similar to the situation during contraction by a myofibril. Let us now view the molecular detail of the actin and myosin proteins to demonstrate molecular events that occur during a sarcomere contraction.
And then we'll watch the next one again. Or we'll watch the next one next.
This scene shows the molecular relationships of actin and myosin relative to the banding pattern of the sarcomere and demonstrates how this relates to sarcomere contraction.
The blue things here are the Z lines.
Note the relationship of actin and myosin molecules relative to the banding pattern that we have discussed for the sarcomere. Myosin filaments consist of crosslinks that bind to specific receptor sites on the actin. The crosslinks pull the actin along the myosin.
Notice the kind of ratcheting action.
Note that the crosslinks attach and release independently. If the crosslinks acted in unison, they would all be released at once, and the myosin would be detached from the actin, allowing the sarcomere to go back to its relaxed, uncontracted state. Let us look more closely at how the crosslinks slide the actin along the myosin.
I put together two.
One more
This scene
video.
shows the events that activate muscle contraction and the biochemical details of cross-linking and sliding of the actin filaments by the myosin heads. The cycle starts when an electrical impulse from a motor neuron stimulates a muscle fiber. Invertebrate animals may have 1, 2, or 3 motor neurons that control the contraction of a single muscle fiber.
Don't worry about that
And
detail.
each motor neuron has multiple end-plate junctions spread over the fiber surface. The nerve signal from a motor neuron does not generate an action potential in an invertebrate muscle fiber, but rather depolarizes the entire surface of the fiber to produce contraction.
And again, that doesn't apply
The
to
electrical
humans.
depolarization also spreads into tiny membranous invaginations of the fiber membrane called transverse tubules or T-tubules. The T-tubules extend to the myofibrils. The
So
T-tubules
this does
align
apply to
with the
humans now.
A-band in invertebrate muscles. The T-tubules essentially carry the electrical signal into the sarcoplasm and along the sarcoplasmic reticulum, a complex of membranous vesicles that serve to store calcium within the muscle. Depolarization of the T-tubule membrane activates calcium release channels of the sarcoplasmic reticulum to release stored calcium into the sarcoplasm. The released calcium then initiates the muscle contraction by binding to a protein called troponin. Calcium-activated troponin causes a conformational shift in the protein tropomyosin, which winds around the actin helix and blocks the myosin binding sites on the actin molecules. This frees the actin molecules to bind the myosin head unit. The contraction cycle starts with the myosin head bound to adenosine triphosphate, ATP. The myosin head contains adenosine triphosphatase, an enzyme that splits the high-energy phosphate bond of ATP to produce adenosine diphosphate, ADP, and inorganic phosphate. The resulting ADP and inorganic phosphate remain associated with the myosin head, which is also charged with the energy released by the splitting of the high-energy phosphate from the ATP. To start the contraction cycle, the myosin head attaches loosely to actin, then releases the inorganic phosphate, which causes tight binding with the actin. After tight binding, proteins in the myosin head undergo a conformational change that results in repositioning the angle of attachment between the myosin head and the actin. The shift to the new conformation pulls the actin along the myosin. After pulling the actin, myosin exchanges ADP for a new molecule of ATP. This results in release of the myosin head from the actin. The myosin head resumes its relaxed conformation and position, and the cycle is ready to repeat. Let us watch the cycle repeat several times.
So the attachment of the myosin head to actin is called cross-bridge formation, as per our textbook. And notice that the ATP attachment to the myosin head is required for release or breaking of the cross-bridge formation. Okay, let's get back to our lecture. So I'll post the links to these 3 videos I just showed you, because I managed to pause it for a few seconds in between, I think, the second video. So this process of the filaments sliding past each other, thin versus thick, is called the sliding filament theory of sarcomere contraction, muscle contraction. The Z lines get pulled closer to each other. Where's my cursor? Here. As you notice here, during contraction, the A band remains constant, because that's the dark band that has just thick filaments. It appears constant, it appears dark. The H band will disappear with full contraction, because with full overlap of thin and thick filaments, that H band gets squeezed out in a way. The I band also decreases, not fully, but it'll get shorter, as you see here and over here. Okay, so let's talk about the neural control at the neuromuscular junction. So the motor neurons, this is the cell body of this neuron. It's in your spinal cord. It has a long extension called an axon, and where it reaches into the muscle and the fibers, it branches, and it ends at what are called axon terminals, where the neurotransmitter acetylcholine is stored. So these motor neurons stimulate muscle contraction. In your brain, you have neurons that generate an action potential. Those neurons connect synapse with neurons in your spinal cord. They synapse with this or these motor neurons, and the action potential shoots all the way down to the axon terminals. So that's under conscious control. Okay? And each muscle fiber is controlled by one motor neuron, and each motor neuron controls multiple muscle fibers, as we'll get into. But here we're just showing a one-to-one relationship. And then the motor neuron innervates the muscle fiber at the neuromuscular junction. If you remember, the neuromuscular junction is the axon terminals of the motor neuron and the modified sarcolemma, called the motor end plate, of the muscle fiber. You can kind of see here the motor end plates very convoluted to increase